authorities struggle to respond to the COVID-19 viral pandemic, everyday life is drastically altered by travel restrictions, school closures, and diminished business and social interactions.
It is the most extensive lockdown the world has ever seen.
But even as much of our world appears on the verge of completely shutting down, some among us jump into action. From the outset of the outbreak, the scientific community quickly geared up for an urgent battle.
Each pathogen has its own path that it takes in that context, and this one has surprised many of us in terms of the speed with which it spread through the Chinese population, and then subsequently the speed with which it has impacted other countries more globally.
This is a disease that is completely new, so we don't know really a lot. Every day that goes by, we learn more. At the same time, this is a very fast-moving target, and so the more data we have, the better it is.
And that knowledge forms the backbone of a critical quest to develop a vaccine to help defeat COVID-19. The Coalition for Epidemic Preparedness Innovations, or CEPI, supports vaccine research around the world. But the transition from theoretical endeavors to real-world needs happened more quickly than anyone anticipated or hoped for.
This is the most frightening disease I've ever encountered in my career, and that includes Ebola, it includes MERS, it includes SARS. And it's frightening because of the combination of infectiousness and a lethality that appears to be many-fold higher than flu.
But despite the urgency of the COVID-19 pandemic, epidemiologists and government health officials must face a harsh reality. Vaccine development and testing takes time, at least 12 to 18 months.
The vaccine is quite down the road at this point. And now there are a lot of programs are starting worldwide to find those weapons to fight the disease.
We'd be giving this to normal people to prevent infection. So you must be sure the edict of medicine, first do no harm. So we need to make sure it's safe, and we need to make sure it works. That entire process will take at least a year and a half.
Professor Paul Young leads one of the most promising research operations at the moment, with his team at Australia's University of Queensland, thanks in part to a CEPI-funded slight head start. For the past 6 years, they focused their work on broad-based vaccine modeling, using mRNA coding, an adaptable methodology able to quickly respond to new viruses as they emerge.
So we've developed a generic platform technology for vaccine production that could be applicable to a wide range of viruses, and that's why we've been able to apply it to this previously unknown virus relatively rapidly.
Amid the initial escalation of the public health emergency in the city of Wuhan, the Chinese released the genetic sequencing of this new coronavirus, COVID-19, on January 10th of 2020.
And as soon as that was done, we began work on developing our vaccine approach to it. We made more than 200 different versions before we settled on one particular vaccine candidate, and that has already gone into animal studies to prove that we actually induce an immune response that is likely to protect animals. Once we've done that, and we've also gone through toxicity studies, then we'll hopefully be ready to enter clinical trials, human clinical trials, in late June.
COVID-19's spike-like structures exhibit a specific protein to gain entry into its host cells in order to replicate.
And one of the prime immune responses is an antibody response that targets that particular protein on the surface of the virus. And that can generate a neutralizing activity or a killing activity that destroys the virus.
Young's line of attack aims to unleash the body's defenses by introducing this protein, stabilized or clamped in position as it would appear on the virus's surface.
so that when we vaccinate individuals, their immune response sees this protein as though it was the virus itself. And they will mount an immune response that can then become their memory response if they subsequently encounter the live virus. So we're trying to mimic the outer exterior of the virus by generating this particular protein as the vaccine immunogen.
Disease pandemics litter human history. But vaccination technology is a relatively recent tool in our medical arsenal, one that arose kind of by accident, a response to smallpox in the 18th century, thanks to the discriminating eye of Dr. Edward Jenner and others.
People used to say, find a milkmaid, she'll be the most beautiful girl in town.
Jenner deduced the women's work impacted their appearance.
In putting two and two together, the reason they never got smallpox is because by milking their cows, they got blisters on their hands, which were called cowpox. And getting blisters on their hands prevented them, immunized them, we would now say, from getting smallpox.
So he created the first vaccine, a thread soaked in a concoction of cowpox material, and exposed it to patients by cutting a small slit in their arm.
And it wasn't until almost 100 years later that we began to understand that there was such a thing as an infectious disease, and that the reason people didn't get most infectious diseases twice is because they were immunized.
As scientific understanding grew, so too did the use of vaccines to do battle against some of our most feared diseases, like polio.
The parents of 440,000 grammar school children in 44 states gave their permission.